Miss Ashman, could you give us an idea of how many students are on YouTube when you have a chance, please?
Are presenters not on as yet, though?
No, they're not. I'm just checking on the YouTube side.
Okay.
Ask the coordinators if they could just send the reminder to the student.
What was that, Dr. Monroe?
Yeah, that was for me. That was for me.
Oh, okay.
Thank you for your quick ears, Miss Walker.
Good afternoon, Dr. Carrington.
Hi, good afternoon. How are you?
I am fine, thanks. Good to see you and have you.
Oh, it's a pleasure. Let me get them so you can actually see me.
Yes.
All right.
Okay.
So, and you're hearing me very well and everything?
Yes, we are hearing you clearly.
Okay, excellent.
Right.
Hi,
Hi,
Dr.
hi.
Carrington. So I'm Melissa Walker. The last email you would have gotten would have been from me.
Thank you.
You're welcome.
Nice seeing you.
Yes, good to see you too.
All right, so we're waiting on a few more students to join the session. They're joining the YouTube session.
Okay.
Yes.
So let's give them about another 5 minutes.
Okay.
All right, great.
And we could wait for the other presenter too.
Yes.
Just so we have both of them on.
So how many students do you have?
We have, go ahead, Melissa.
In the school, over 600.
Okay, but how many are you expecting today?
Well, actually we're expecting, and that's just the Mrna cohort in WJC. So Mrna WJC together, it's probably about 800.
Okay,
and then we had extended an invitation to our tertiary level institutions. We have 3 of them in Jamaica and 2 overseas. So we are expecting quite a lot of that's why we opted to have the YouTube link, so that it would open up the opportunity for more persons.
Well, I hope my content is appropriate. I thought I was doing more of a lecture, so I hope it's appropriate. So I thought of it as a teaching session, so I hope that's
going to be fine. That's going to be fine,
because I'm taking them from basics all the way up, you know.
Yes,
and I'm sure they'll appreciate it.
Okay, virology 101 and everything. Okay, great.
Okay.
So we are awaiting the next presenter before we begin, or we could just go ahead.
We could, Mrs. Anderson Johnson, we could wait just about 5 more minutes and then possibly begin.
How many students are there now?
13, so we do have quite a way to go, read the students. And this is really for them.
Okay.
So Prof. Carrington, if you wouldn't mind just waiting a few more
No problem
to
at
have
all.
as many of them join as possible. I'm trying to see if I could reach out to the coordinators to have them hurry them along.
Oh, no problem. No problem.
Okay. Okay, I think it is now 5:40. Yes, my microphone is on, and I hope you're hearing me clearly and loudly enough. It is now 5:40, 10 minutes after start time. So I hope we have the majority of students in the link, and we are going to start. So I'm going to ask us first to just pause. Let us pause and quiet our hearts. We can close our eyes if we want to. Dear God, we are the love of your life. We pray that you keep us safe in your arms. Help our work this afternoon, and may we feel the presence of your Holy Spirit. Amen. Okay, thank you. Welcome everyone, all our colleagues. Welcome to our presenters. Welcome to our students. Students, a big welcome to you. Yes, this is another Zoom session. And we're here to learn a lot. So my name is Pauline Anderson Johnson. I'm a lecturer at the UWI School of Nursing in Mona, and I will be your moderator this afternoon. We will be here for about an hour and 15 minutes, and we'll hear from two key presenters. I'll tell you more about them in a while. We hope that when you leave this session, you will have increased knowledge of the COVID-19 vaccines and their development, and an understanding and appreciation of the process of the rolling out of a new vaccine here in Jamaica. We are going to first of all have Dr. Carrington as our presenter, and then Miss Dawn Marie Richards. But first, our head of school, Dr. Monroe, is here, and we're going to invite her to bring greetings.
Good afternoon, everybody. How una do? How are you doing? I'm in the Zoom platform, but I'm streaming to YouTube. So there are things that I'm able to do now that I was never able to do before COVID. Now I can Zoom and I can stream. Oh, I'm telling you, I think I'm soon going to become a hologram. And welcome, 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 everybody. Welcome, students, especially. You are our guests of honor. I would begin by thanking Mrs. Cheryl Garrix Lloyd for taking the initiative to search Jamaica and the Caribbean for the best possible persons to share in this forum with our students. So we have Miss Dawn Marie Richards, nurse extraordinaire, and the Professor Christine Carrington, who is a virologist. I mean, I read her bio and I am just blown away. We recognize that while public health, public education campaigns have been directed towards sensitizing the population to the COVID-19 vaccination process, healthcare workers require a deeper level of engagement, given our primary patient care roles and our baseline knowledge of the biological sciences. Nursing students are in a unique position, and not just nursing students, medical students, pharmacy students, all students of the healthcare. And we hope that we have a mixture of students here this afternoon. So healthcare science students are in a unique position of transitioning from being a regular member of the public to being a healthcare professional. Hence, there is need to cater to the educational needs of this unique group. Earlier in this year, the Chief Nursing Officer Jamaica, under the aegis of the Ministry of Health, hosted a student forum that was aimed at engaging students in discourse regarding the vaccination. While members of faculty have been capable of fielding students' queries related to programmatic issues, when it came to matters related to the virus, and to our experts in here, our students can ask some tough questions. So when it came to, what is this virus about? How does it work? How does the vaccine interact with our biological systems and so on? We can hazard a good guess, based on our own backgrounds, but we couldn't speak with the same degree of confidence as someone who is an expert in the field. We've been bombarding our resident public health nurse, Mrs. Chisholm Ford, who is also a member of our faculty. And we decided, it was time for us to get some other engagement. So it was necessary to bring in these experts who are with our students. It is our hope that students and the supporting audience, also known as the lecturers and admin team who have joined, will benefit from this initiative. I'd like to thank our presenters, Prof. Carrington and public health nurse Dawn Marie, my namesake. Thank you so much for sharing your knowledge and your time with us. We're honored to have you. Thanks all.
Thank you very much, Dr. Monroe. And just a reminder, 16 months ago, one part of the world woke up to this new disease, which soon became a pandemic. And about 13 months ago, I think Jamaica diagnosed its first case of COVID-19 disease. We have been in the pandemic and it continues. If we look at our public health and our epidemiological history, we will note that vaccines have saved millions of lives each year. And according to WHO, who did a very recent release, it says, quote, COVID-19 vaccination campaigns have officially been rolled out in over 100 countries and are providing hope. And I want us to rest on that word, HOPE, hope, right? So although the pandemic continues and more work needs to be done, there is hope. Okay, and today we are going to hear from Dr. Carrington, first of all. And let me just tell you a few things about Dr. Carrington. She's a professor of molecular genetics and virology in the Department of Preclinical Sciences in the Faculty of Medical Sciences at the University of the West Indies, but the St. Augustine campus in Trinidad and Tobago. She did her BSc in biotechnology, so we can see how she's developed in this area from King's College, University of London. Then she did her PhD in molecular virology. And she remained in England and did her postdoctoral research until 1996. Then in 2004, she was the recipient of a Commonwealth Research Fellowship at the University of Oxford, where she received training in virus evolution and phylogenetics. Her primary research focus is understanding evolutionary and ecological factors that are involved in the emergence, the spread, and maintenance of emerging viruses. She is really more than capable of addressing us today. She is the current head of department of the preclinical sciences at the Faculty of Medical Sciences in St. Augustine, and she's a former deputy dean. She's also the current director of the Global Virus Network Affiliate Center, which is in Trinidad and Tobago. She is part of the UE COVID-19 task force and the Technical Advisory Group on Immunization in Trinidad and Tobago. She is the principal investigator for the COVID-19 Impact Project that established a local capacity for the SARS-CoV-2 genomic surveillance in support of the COVID-19 mitigation and control efforts of local and regional public health bodies. Dr. Carrington, we welcome you this evening, and you have the floor for your presentation.
Thank you so much. I'm really pleased to be invited today and looking forward to hearing questions at the end of my talk. What I'm going to present is a sort of, I'm starting from the beginning with, you know, as simple as what are viruses, and then I'll move along to how vaccines work. And then I'll talk a bit about variants and what that means for the vaccine programs that we have. So we're going to start with some 101. We'll talk about COVID-19 as well, but just that might be a bit of a refresher for some of you because we've been living with this thing for so long. So let me share my screen. Yes, now get my presentation up. Hold on. All right. So you should be seeing my slide now. Let me make sure is that what you're seeing now? Just my slide. You're not seeing my, you're not seeing any, you're seeing one big slide, right?
Yes.
Right, yes. Great. Okay. So we're going to talk about the viruses. I suppose it should be called viruses, vaccines, and variants. And as I said, I'm going to start right at the beginning. But first, let's just orient ourselves. First of all, let me get my pointer. 1 second. Right, so let's orient ourselves in this pandemic that we are having to live with. So first of all, as you well know, SARS-CoV-2 is the virus that causes COVID-19, and it is now responsible for over 140 million COVID-19 reported cases around the globe. Now, this number was from, I think, 2 days ago, one or 2 days ago, 1 day ago. And we've also seen about 3 million deaths around the world, and that's reported deaths. The impact of the number of actual cases is obviously going to be larger than the ones that are actually documented. The virus is spread, as we know, by close contact between infected individuals. It's spread via respiratory droplets primarily. And over the past year, we've seen some amazing scientific advances that have led to us having a much better understanding of the virus, how to best detect it, how to treat it, how to prevent, to treat COVID-19 disease. And most importantly, now we have several safe and highly effective vaccines. So in a very short time, we've come a very long way from knowing nothing about a virus to having a vaccine. If you think about something like HIV, it took 2 years. I mean, with SARS-CoV-2, with COVID-19, it took about a few weeks to figure out what was causing the disease. And when HIV first emerged, it took 2 years to work out what virus was causing the disease. And up to now, we don't have a vaccine, okay? So we've made some major advances. Now, as I said in this lecture, what I'm going to do is provide an overview of what we currently know about SARS-CoV-2. I'm going to introduce you to aspects of virology so that you have a sort of basic understanding, so that you can understand how vaccines work and what these variants are that have emerged in the past few months. And some of it, as I said, may be a bit of a refresher, as I expect that by now you're familiar with things like how the virus is transmitted and how we interrupt transmission, etc. But it doesn't hurt for us to repeat it. So we'll start off with our virology 101. So viruses are tiny infectious particles. And the key thing about them is that they can only replicate inside of a living cell. This is very different from things like bacteria. Bacteria can replicate in the soil. They can replicate in food. They can replicate on your skin as well. They can replicate in your gut. The viruses, they must be inside of a living cell to replicate. And because of this, they're referred to as small obligate intracellular parasites, okay? Now, they're very simple, deceptively simple. They consist of some sort of genetic material, which is shown by these two lines here, that is surrounded by a protective protein coat. And so for many viruses, that's all it is, genetic material and a protein coat. There are some viruses, however, that also have an extra layer, a sort of envelope. It's a lipid layer. So it's sort of like an oily or waxy sort of bubble around the virus. And in that envelope, as it's called, you have these proteins, these viral proteins sticking out, which we'll come back to again in a little while. Now, the viral genome itself, unlike us, we have DNA for our genetic material. For viruses, they can have either DNA or RNA. All right, so it can be one or the other. They don't ever have both. So they have choices for their genetic material, and different types of viruses will have different types of different genomes. So as I said, we have the envelope, and this complete virus particle is called a virion, okay? So that's one virus particle, that's a virion. And the job of a virion is literally to deliver the virus's genetic material into a target cell, where that genetic material sort of reprograms the cell. It sort of offers its genetic material to the cell. And the cell, instead of doing its normal work, will look at that genetic material and start to replicate it. And it will start making new virus particles. So the job of the virion is to deliver the genome, that's the full complement of genetic material, into a host cell, where with the help of the host cell machinery, it can make new virus particles. So the viruses sort of hijack the host cell and use its resources to make more viruses. So basically reprogramming the cell to be a virus factory. Now, because they can't reproduce by themselves, sometimes viruses are not even considered to be living things. There's a big debate about that, whether they're alive or not. They don't have any cells. They're very, very small, much smaller than the cells in our body, much smaller than bacteria. Okay, so they're very peculiar organisms. Now, as I mentioned before, the genome of the virus is basically its full complement of genetic material. And as I said, it can either be DNA or RNA. And I don't know if you all remember that DNA is a double-stranded molecule. So this is what DNA looks like here. And this is what RNA looks like. It's a single-stranded molecule. It has a slightly different basic units. But what's important is that both DNA and RNA, in their structure, they have a sort of regular backbone to their structure. And then projecting outwards from that structure, you have these, what you call nitrogenous bases. And I'm mentioning these because we're going to talk about variants and mutations and stuff. So these bases, these nitrogenous bases that stick out, there are 4 of them that are found in DNA, cytosine, guanine, adenine, and thymine. And they are, you can abbreviate that as A, T, C, and G. And in RNA, you have cytosine, guanine, adenine, and a different one. Instead of thymine, you have this one called uracil. So you have C, G, A, and U, and they make up the they make up sort of sentences that carry information, instructions for how to make new viral particles. So this figure here, don't be scared of it. This is basically the SARS-CoV-2 genome. It's made out of RNA and it's 30,000 letters long. And this is just a small part of it, actually. So this is actually a small part of the genome of the original virus that was isolated in Wuhan in China. So you can see it's just a sequence of these units called nucleotides that are strung together, and each of those nucleotides has a nitrogenous base. So what we're reading out here is the sequence of nitrogenous bases. Okay, so that's this. When we talk about sequencing a virus's genome, this is what we're doing. We're working out that sequence. And this sequence encodes how to make proteins, viral proteins. Okay, now viruses come in all shapes and sizes. And this figure here just shows the structure of viruses belonging to, they have a whole wide variety of families that they belong to. And this just shows a whole bunch of them. All right, so they look very different, but generally at the heart of them, there's genetic material surrounded by a protein coat, and in some cases surrounded by an envelope. So some of them have DNA as their genomes. These are all DNA viruses. RNA as their genome. These are all RNA viruses. And I've put this blue line around the ones that have envelopes. And here is the family that SARS-CoV-2 belongs to, the coronavirus family, okay? So that's where this is the culprit, belongs to this family here, but there's a whole, there are thousands and thousands of different viruses. Now, in everyday life, when we think about having a virus, I don't know if you all say the same thing in Trinidad, but Trinidadians are constantly saying that they have the virus, which could be any virus, okay? So when somebody has the virus, we tend to think of it as a collection of symptoms that you get when you're infected. So, if you have a flu, you're thinking of fever and aches and pains. The chicken pox, you're thinking of a rash. But what is actually happening in your body when you have a virus, when a virus infects you? So at the microscopic level, what is actually happening is that the viruses are, as I said before, they're using your cells to make more copies of themselves. So while your cell's machinery, which should be spending its time looking after your cell and your body and doing its job, a kidney cell doing its kidney job, liver cell doing its liver job, instead of doing that, what it's doing is making more virus particles. And that causes damage to the cells directly and indirectly, and also damage to tissues, damage to organs eventually. And that is what causes the symptoms that we see, all right? That fact that these viruses are replicating in your body. So this just shows sort of typical virus life cycle. So here is a virus particle here, and as I said, the job of the virus is to recognize and enter a host cell. It reprograms the host by providing instructions in the form of viral DNA or RNA, and it uses the host resources to make more virus particles. So it sort of, gives the cell a computer program, a program to work with, and the output of that program is more viral particles. So the virus attaches to the cell, then it enters the cell. Once it's entered, the protein coat is lost and the viral genetic material is released. That genetic material is replicated, so you get copies of the genome being made. That genome is also used to make viral proteins. And those viral proteins come together with the genetic material to sort of assemble into new viral particles. And then the virus is released and it's off to infect a new cell or to infect a new person if it gets out of your body to the next person. All right. So that's what's going on in the cell. And the tissue, the cell type, the viruses they target different tissues and different cells. So depending on the virus, so like coronaviruses, for example, and the COVID-19 virus, they all target cells that are found in your nasal passages and down in your lungs. So that is why they're associated with respiratory symptoms. But other viruses will target different cells. And this diagram just shows the whole range of different types of symptoms that you can get from different viruses, which is all associated with what type of cells and tissues they infect and how they interact with your body. So you can have meningitis, you can have, sort of common cold type symptom from another virus. There are those viruses which are associated with rashes. We have some which cause gastrointestinal symptoms, etc. Okay, so it all depends on the virus and what part of your body is interacting with. So I just want to turn now to spend a little more time on the actual coronaviruses, and don't get scared by this table here. It looks a bit busy, but we don't have to take in everything. So SARS-CoV-2, the virus that causes COVID-19, is a coronavirus, okay? Now the coronaviruses, they are members of a large and diverse family of viruses, and the whole family is actually called Coronaviridae. They are found in a wide variety of animals, and this is all stuff that we knew before, okay? A lot of people believe that, SARS-CoV-2 is a new coronavirus, but we have experienced coronaviruses before. People were doing research on coronaviruses before. We know that they're found in a wide variety of animals. And these animals, what I've shown here is a whole range of different animals that harbor different types of coronaviruses. All of these coronaviruses that we find in the different animal species, they are respiratory viruses. So they cause a range of illnesses from mild colds to severe pneumonia. And that's in the animal hosts that they normally infect. And we're learning from SARS-CoV-2 now that there can be a range of other pathologies associated with coronavirus infection. So we always thought to them about, your respiratory viruses affecting your lungs and your sort of nasal passages and stuff. But now we know they cause blood clots. Now we know they can cause your fingers and toes to turn, dark. They affect your cardiac tissue. Some of them can affect, have neurological effects, etc. And we're learning all of that because this massive pandemic that we're going through is giving us there's probably certain things that occur at very, very low frequencies. But because of the massive number of people who are being infected, even those very rare outcomes, we are able to observe them. So things we would not have seen before in other coronaviruses, we're seeing here. Now, the coronaviruses are divided into 4 large groups. And I've highlighted here those coronaviruses that are known to cause disease in humans. And there are actually 7 of them that we know of. The ones in blue that I've shown here, we've known about for a good while, and they are actually associated. They circulate in the environment all the time. They're known as seasonal coronaviruses, and they're amongst the viruses that are associated with common cold. So if you've had a cold in the past, I think it's about 15% of sort of influenza-like illnesses in the past would have been due to a coronavirus. So you may have had a coronavirus infection before, different coronavirus, obviously. And the ones that I'm showing here in red, these are ones that emerged from animal populations into human populations within the last 2 decades. So they are all associated with, so they're new to the human population. And they're all associated, have all been associated with epidemics of severe disease and death. SARS coronavirus was active in about 2002, 2003, and I think it only caused about, now we thought it was a lot then, I think it was 8,000 deaths or 8,000 cases, which we thought was a lot until we met this next one. We have SARS-CoV-2. MERS is very similar. It is found mainly in the Middle East. It's called Middle East Respiratory Syndrome coronavirus, and that's spilled over from bats to camels and from camels to humans in the Arabian Peninsula. And then we have SARS-CoV-2 here, which has gone on to cause the major pandemic that we're living through now. So coronaviruses are not new, okay? This one just appeared, and these 2 appeared in the last 20 years, but were contained. Now, I'm not going to spend too much time on this, just to remind you that the majority of people who get infected with the SARS-CoV-2 recover, without any medical intervention. So about 81% of cases are mild. There's also a large percentage of cases that are thought to be asymptomatic. The people don't show any symptoms. And what I want to just drive home here is the fact that this, and I think you would be aware of this, we have this sort of, majority being mild cases, then it's about 14% severe, and then, 5% being critical. And then, of course, you always have, unfortunately, deaths from this virus. So it is a deadly virus. The vast majority are mild. What we have to remember, though, is that this risk, this pattern here is not evenly distributed across the population. As we know, people who are the older people have a much higher risk of severe disease and death, and certain underlying health conditions can also increase your risk of death. So it's not evenly distributed. Transmission, I think we're all familiar by now with the mode of transmission of SARS-CoV-2. It is spread by respiratory droplets that are expelled when an infected person speaks or coughs or sneezes or laughs or shouts, and it comes out of their nose and mouth. Now, in order to infect a person, the virus has to get inside your body. Remember, this virus it has to get into your body and into a cell. And it has to get in through an opening. It can't penetrate your skin. A lot of people in the early times were very worried about, just touching something is going to infect me. No, touching something is not going to infect you. You have to introduce it into your mouth, your nose, or your eyes. Okay, so it has to get in through an opening that leads to your respiratory tract. Okay, so people can either get infected directly, all right, through aerosol droplets if the droplets from an infected person enter their nose, their mouth, or their eyes, or you can get infected if you touch a contaminated surface and then touch your nose, your mouth, and your eyes. Now, what I want to mention here is that the major route is via these droplets. We have spent a lot of the last year sort of cleaning madly, sanitizing everything, which is good. However, the vast majority of transmission we have learned occurs via this route here. So it is not about, it's really about close contact with infected individuals. So if you had a choice to give up one of the things you're doing, not that I want you to give it up, but this would make a much less impact, cutting down and cleaning, than this. Wearing a mask and social distancing is the key, okay? Because that is where the vast majority of transmission is occurring. And I've just put this gentleman here to show you that, people get very tied up with, oh, somebody looks sick, they're coughing, they're sneezing, I can get COVID from them. Don't forget, viruses can be transmitted by people who have no symptoms, okay? So perfectly healthy. In fact, most people are infectious 2 days before they start to show symptoms, before they feel their first ache and pain or fever or headache or whatever, they are already infectious, okay? So of course, wearing masks and physical distances reduces the risk of droplet transmission. We are well aware of that. And yes, so this was just to mention that the direct and indirect contact was a lesser method. In terms of the virus's survival in the environment, just to say that there are a variety of things in the environment that can inactivate the virus, heat, humidity, all these things, and help to decay, help the virus to break down basically and become non-infectious. But the rate at which that occurs will depend on the type of virus it is and the environmental conditions. Luckily, coronaviruses are easily inactivated by standard disinfection methods. So simple things like washing your hands. People laugh because they say you have this terrible killer virus and you're telling me that to protect myself, I should wash my hands. Remember I told you that virus has this sort of oily, waxy envelope around it? Well, that oily, waxy envelope is totally necessary for that virus to infect a cell. And what we know, for example, soap and water, that destroys, soap, detergent cuts through grease. And so that same oily envelope is going to be destroyed by hand washing and things like that. So it works well. So just to emphasize, no one intervention is completely protective. So these are all the things that we're used to. And now we can add vaccines, okay, to protect us from COVID-19. Right, so now I want to turn to about vaccines and how they work. So let's turn to the immune response to viral infections, just to provide some background to how these vaccines work. So as we said before, when an infection occurs, the body, the virus goes into the cell and it starts to replicate. So this is an infected cell here, all right? And what happens is when you have an infected cell, this cell is busy making new viral particles, and a lot of the viral proteins are sort of displayed on the outside of the infected cell. So you have a cell with all these proteins that are foreign on its surface. And then of course you have viral particles moving around, for example, in the bloodstream or the intracellular fluid and stuff like that, going from one cell to the next. So when this infection occurs, your body recognizes the virus particles and the viral proteins that are on these infected cells. It recognizes them as foreign invaders and it mounts an immune response. Now, the immune response involves the immune system sort of sizing up this invader and then selecting and making specific tools to combat that threat, okay? And those tools would include specific immune response cells called B cells. And the job of B cells is to make these proteins called antibodies. antibodies sort of recognize and bind to the virus particles. So they'll bind to these virus particles here and block them from infecting a cell. Some antibodies, they can also bind to the infected cell. And when they bind to the infected cell, what they do is they sort of act as a signal for another arm of the immune response, these T cells, to now recognize these infected cells and they make other T cells. So you have different types of T cells. Some of them kill infected cells, and others, they stimulate B cells to make antibodies. So we have these two arms. Now, what's very important is that when the immune system, so it sees this invader, it makes all these tools and it starts to fight against the virus. And when the immune system wins, you clear the virus and you recover from the disease. Now, importantly, the immune system remembers what it has seen before. So it remembers viruses that it's seen before. So if it encounters the same virus again, it can go into action much, much more quickly than it did the first time, and either prevent the infection from taking hold at all, or at least preventing it from causing illness, or preventing and dampening down that virus to the extent that you're not going to spread it to other people. So when you think about, it's a reason why we only get measles once. It's a reason why you get chickenpox once, because your body mounts an immune response. Yes, you get some symptoms, you get a rash with chickenpox, you get fever, but at the end of the day, most of the time, your immune system wins and you get rid of that virus, you recover. And then the next time, it's not that you're never ever exposed to chickenpox ever again, but the next time you meet chickenpox, your immune cells are going to say, I've seen this before, and they can just draw the tools that they need out of their toolbox to fight immediately, and they will get rid of the virus much faster, okay? So the immune system has memory, so it remembers. So the main job of the body's immune system is to recognize invaders, to fight them off before too much damage is done. And when the immune system wins, recover from the illness, and they remember the foreign invader so that next time it comes, they can deal with it much faster. So now we've absorbed that, we can now turn to vaccines. Right. Now, when you're infected, yes, you produce an immune response, and sometimes the immune response wins, right? But in the time when, during that time when you've been infected, you can get very, very ill, as we've seen with people who get COVID-19. People are hospitalized, people's lungs are damaged, they have long-term problems, etc. Yes, at the end of the day, they're immune. Yes, I had measles, and some people recover from measles. There are people who die from measles. And measles is not a pleasant illness to have. Nobody wants to be ill. So vaccines, what they are biological preparations that basically train your immune system to recognize and combat specific infectious agents by triggering an immune response without causing disease. So this is wonderful. We can get those memory cells, we can have those tools at hand. Your immune system can be made ready to respond immediately to an invader, because we have tricked it with this vaccine into believing that it's seen this virus before. So you're basically, put simply, a vaccine is a preparation that tricks your body into believing that it has been infected by presenting it. And it does this by presenting it with either similar or modified virus, or with proteins from the disease causing target virus. Remember I said that the immune system recognizes these proteins from the virus. So when you make a vaccine, there's three main ways in which this can be done. I can take a whole virus particle. So if I want to protect against virus A, I can take virus A and I can modify it in a way that I prevent it from replicating. Let's say I treat it, I kill it, basically. And I use that as my vaccine because the proteins are still there. The body will still recognize it, but if it can't replicate, it can't cause disease. So that's one way. Another way is to take those proteins, these pink proteins sticking out on the outside. Another way is to take those proteins and purify them. So you forget all this stuff with the genetic material and the rest of this code here. Let's just take these these proteins from the virus, and we put those into your body, right? So that's your vaccine. And the immune system sees those proteins and said, oh my goodness, I've been invaded by virus A, and starts producing these, mounting an immune response. The other clever way to do it, which is where we get these mRNA vaccines, and there's a reason for doing this, is to introduce the genetic material from the virus. Now, the genetic material carries instructions for how to make viral proteins. It carries instructions for how to make an entire virus particle. So what we do is we take a part of the genetic material, not the whole thing. We take a small part that is used to make a specific protein that we know is going to produce an immune response. And in the case of SARS-CoV-2, that protein we use is called the spike protein. So we take this genetic material that encodes the spike protein and we introduce that into the cells of the person you want to vaccinate. And the reason why it is clever, I mean, you could just take the proteins and put them in, right? But remember, when the immune system responds to an infection, what it is doing is responding to both viral particles, so particles, and to infected cells, okay? So it's recognizing the sort of particulate virus in the thing, and it's recognizing virus in the cells. When you use just subunits like this, you don't necessarily stimulate the immune system entirely. You tend to stimulate more of a B cell than a T cell response. If you use a vaccine that mimics the way the virus behaves, so you just literally put it in this piece of genetic material into the cell is going to produce those proteins you want, display them on the outside of the cell, and you get both a good robust B and T cell response. So there's reasons for using different types of approaches. So this is just the same thing here. Remember I told you could use the whole virus. So, but of course, we don't want to use a whole virus that's going to cause you disease. We want to use a whole virus that's not going to cause disease. So you can take, as I said, the virus that you want to vaccinate against. So we could have taken, for example, SARS-CoV-2, and we treat it with, weaken it somehow. So it's called a live, but it still replicates, but it can't cause disease. It's sort of weakened. That's a live attenuated virus. Otherwise, you can take the virus and just knock it stone dead. You treat it with chemicals and it's not going to replicate, but the proteins are still there. So that's called a dead or inactivated vaccine. You can say, well, I'm not even going to put SARS-CoV-2 inside of this person. I'm going to take another harmless virus and I'm going to modify it so that it produces SARS-CoV-2 proteins. It's not going to cause disease, but it has those pink proteins on the outside. So we're going to get a SARS-CoV-2 immune response. In this case, it's actually displaying the proteins, and then you have these viral vector vaccines. This is actually one that is used for Astrazeneca, where you have the genetic material here, and the virus, this particle, is just acting as a vehicle to deliver that genetic material into your cell, which will then make the proteins. You could, as I said, you could use viral proteins that the immune system recognizes, and there's two ways to do that. You can purify the proteins. There's some SARS-CoV vaccines, some COVID-19 vaccines that work this way. It's a purified subunit. Sometimes, or alternatively, you can take the proteins and sort of put them into a sort of virus-like particle. There's no genetic material or anything, but it sort of mimics the virus more. And then, of course, we have the DNA and RNA-based ones where you use the genetic material alone, which is what I mentioned before. So just to look at some of the COVID-19 vaccines, there are many, many COVID-19 vaccines. Many of them have been approved for use, and there are many, many more in the pipeline. And these are just some of the main ones we are most familiar with. So we have the Oxford Astrazeneca vaccine, which is being used throughout the Caribbean. This is provided by the COVAX facility. It's a viral vector vaccine. So what it is a virus called an adenovirus, which in this case, it's a virus that normally infects chimpanzees. It causes common cold in chimpanzees, but it causes no disease in humans. And what they've done is they've modified it in two ways. One, so it cannot replicate, and two, so that it carries the gene for the spike protein from SARS-CoV-2. So it enters cells, but it can't replicate in human cells. It stimulates a good T and B cell response. It can also be stored. It's in a fridge, and it's cheaper than some of the other approaches. There are a number of other viral vector vaccines working similarly, also using adenoviruses as the sort of vehicle for carrying that genetic material. The Sputnik vaccine works that way, and the Johnson & Johnson vaccine works that way. And then, of course, we have the mRNA vaccines, which just the genetic material, a small piece of genetic material that encodes the spike protein from the virus, and it's sort of enclosed in a lipid bubble. And those are similar, very good T and B cell responses, and they're quick and easy to develop, and they're a little more expensive though. Okay, and we know they're very effective. Now, this slide, I just put this up. This is just some of the vaccines that are approved to date. And we can see Oxford AstraZeneca here, Moderna, Pfizer, etc. And a lot of people get tied up with the what we call the efficacy of, well, it's an important concept, the efficacy of the vaccine. So when you test a vaccine, one of the last stages of testing is what you call a phase 3 trial. And in those trials, you give the you get volunteers and tens of thousands of volunteers, and half of them are given the vaccine and half of them are given a placebo or some sort of like a fake vaccine, basically. And they're not told what they're given. They don't know. In fact, often the researchers who are vaccinating them don't know either. And these people go out into the world, and some of them will get infected and some of them won't get infected with SARS-CoV-2. One of the major reasons we are able to develop SARS-CoV-2 vaccines or COVID-19 vaccines so quickly is that we could get those results very quickly about who was infected and who was not, because when you went to somewhere, test these vaccines in the US or in Brazil or in South Africa, where they had very high viral circulation, in a very short time, in a few months, people get infected. And what you do among all your volunteers, you look to see how many of the vaccinated people were infected versus how many of the unvaccinated people were infected. And the extent to which the disease is reduced in the vaccinated people compared to the people who received the placebo is what you call your efficacy, right? So with Moderna, you saw a 94% reduction in disease, 76 to 68 to 82 with Oxford AstraZeneca, etc. So we have a wide range, and we hear people lamenting the fact that this vaccine only has 56%, and this one has 60, and this one has 98. What we have to remember is that when these clinical trials were carried out, what they were asking the volunteers to do every single day was to tell them, how do you feel? Do you have a fever? Measure the temperature, any aches and pains, headache, any symptoms, anything at all, tell us, right? Every now and again, they would test them to see if they had the virus, right? But they were not tested to check for infection every single day. That'll be too expensive. They were reporting disease. And people would have been reporting mild disease. I had sniffles. Some would be reporting, some of them would be hospitalized. Some of them would have died. And what they found that more people in the people who got the vaccine got sick and died much less often. So this number tells you about the reduction in disease. If we look at these vaccines and we look at the efficacy by the severity of COVID-19 infection, what we find is that all of them are excellent at protecting against death and hospitalization, right? Or severe disease, I should say, is what is shown here. So this is how protect against severe disease. We've seen 100%, 100%, 100% for AstraZeneca, and so we go on. Then we have protection against death, 100%, all the way down. Okay, so when you see, when you're offered a vaccine, I think the best advice is that the first approved vaccine that you are offered is the best vaccine to get, okay? Because even if one of them with lower efficacy is not going to protect you against coughing and sneezing, it is going to protect you against hospitalizations, severe disease, hospitalization, and death, okay? Right, so I'm coming to the end now. Well, a little way off, but we're getting there. Now I'm just going to turn to these variants. So we have these excellent vaccines, several excellent vaccines. And one of the things I should have stressed about the immune response is that the immune response is very specific. So if I get infected with virus A, my body is going to mount an immune response to virus A. It's going to make the right tools to fight virus A. It's going to recognize those proteins on the surface of that virus. If virus B comes and virus B doesn't look anything like virus A, all right, the immune system is, as far as it's concerned, it's never seen this before. This is new, okay, and it has to make new tools. So it's very specific. So what happens to that protective immune response that you get when the virus changes, okay? We care about viruses mutating and evolving and stuff. What happens to the immune response? So we have to look at what happens during an infection. So when a virus is transmitted to a susceptible individual, as we heard before, it enters their cells, it replicates to produce millions of new viral particles. Now, during that process, as I said, millions of copies of the virus's genome are made. So that genetic material is copied millions of times so that each particle that comes out can have a piece of that genetic material inside. Now, one of the issues is that the mechanism that replicates the genome is not foolproof. It does make errors, okay? So occasionally during an infection, you can get errors being made in that sequence, and those errors are called mutations. Now, mutations are normal and they are expected. They are the basis of evolution, okay? So, but you do get errors occurring occasionally. That the enzyme that replicates the genetic material, it's not, 100%. Its fidelity is less than 100%. So this slide here just shows partial genome sequences of SARS-CoV-2 from different patients. And you can see the sequence is identical all the way. They all have the same genome, except that there are few changes. And these changes are the mutations that we're talking about. So you're encoding a protein here, and then you get a little change. Some of those changes will affect the way the protein looks, okay? So over time, right, what's happening? So over time, you get these mutations accumulating, and they sort of give rise to these multiple lineages of the virus, which are basically descendants of the original virus that have these small genetic changes that distinguish them from the original virus and from each other. And so these lineages are like, sort of branches of a family tree. It's the same virus, but slight differences in these different branches. Now, when we hear mutation, or people get scared because we watch a lot of Hollywood movies that tell us that things mutate and then sort of kill us. The vast majority of mutations that occur in these viruses have little or no effect on the virus's ability to cause infection or disease. So the lineages that contain them behave very similarly. However, occasionally you get mutations arising that affect the virus's proteins, affect the virus's properties. So it might affect how easily it spreads, or the severity of the disease it causes, or how well vaccines work against it, okay? So when a virus is widely circulating in a population and causing many infections, the likelihood of the virus mutating increases because there's so many, every cell is making millions of copies of the genetic material. New cells are being infected, millions more, millions of people being infected. So every round of infection is an opportunity for the virus to evolve. And we can use a technique called genome sequencing to sort of detect and track mutations, and genome sequencing studies have shown over the past year that many different lineages of SARS-CoV-2 have evolved with mutations, as I said, that differentiate them. Now, a virus with one or more mutations is sometimes referred to as a variant. So that's a variant of the original virus. So this is where this concept of variants comes in. So just to show you here, this is this is what we call a phylogenetic tree. And it's showing how SARS-CoV-2 has evolved over time. This was between December 2019 and January 2021. So basically, it's a family tree. And here we have we would have the sequence of the original virus. And over time, you get these lineages evolving, different, slightly different versions of the virus evolving around the world. So this just shows how it's happened over time. So at the beginning, all the viruses around the world would have looked very similar. But over time, these new lineages emerge. And if you look at this map here, you'll see that in each country, you're getting a more variety of lineages in those countries. All right, so it expands and evolves. So at the start of the pandemic, all the viruses infecting people were very similar, but over time, they have diverged and branched out like a family tree. Now, lineages of SARS-CoV-2 that have been shown to either spread more quickly or to cause more severe disease, or against which vaccines work less well, are referred to as variants of concern. There are currently 3 major variants of concern if you live in the US that are characterized by specific groups, or what we call constellations of mutations. And there are also a number of other variants that are under investigation or of interest because of the types of mutations that they carry. So this is a tree I showed you here. One of the first variant of concern that was identified is called B117. It was first identified in the UK. So this is it here. This lineage here is what that's B117. Then we have B1351, which was first detected in South Africa. So that's that group there, that lineage growing there. And then we have P1, which was first detected in Brazil. So I'll just say a little bit about those 3. This table summarizes what we know so far about these 3 variants. B117, first identified in the UK. It's found in about 125 countries now, including Jamaica and many other countries in the region. It has 9 spike mutations in its spike protein. South African one is less widely spread, Brazil even less. And they have 5 and 11 mutations respectively. Now, my lab has been doing genomic sequencing of viruses from CARFA member countries. So, you know, what the ministries in the different islands will send samples to CARFA in Trinidad, the Caribbean Public Health Agency for sequencing, and CARFA sends it to my lab. And we do the sequencing and send them back and let them know which variants we're seeing, etc. So, so far, as I said, we've detected B117 in several countries. P1, we just detected in Trinidad. We saw it in Trinidad in a sample from some offshore sample. And then recently on Monday, we saw it for the first time in a local sample. So this is of concern. And then the main concern about these variants is that they contain a large number of mutations. And as I said, including mutations in the spike protein, which is the major target for the protective immune response and the protein around which the COVID-19 vaccines were designed. So that is the concern that we have. Now, laboratory tests have shown that because of these mutations in the spike protein, these variants may be less easily neutralized by antibodies produced in response to infection with other lineages of the virus. So remember, they made all these, people who were infected before would have been infected with a different lineage. Now these variants of concern are slightly changed. So in particular, in the case of the B1351 and P1, you can get reinfections occurring, okay? B117, its immune evasion is minimal. And of course, you also have what we see as a decline in the efficacy of some of the vaccines. In the case of B117, it's minimal, all right? So there's not much decline. In the case of B1351, we see a large decline in efficacy, but very important to remember, this decline is as it relates to mild and moderate infection. So what they're seeing is the vaccine that would have prevented all disease if by other lineages is working well enough to prevent hospitalization and death and severe disease, but not well enough to prevent mild or moderate infection. So that's what we're seeing there. P1 is also, they see reduced efficacy, but not to the extent of B1351. The B117, major thing about it is its increased transmissibility. It spreads from person to person much more easily. For B1351, we don't know yet about its transmissibility. P1 we think is slightly increased, but the percentage of increase to what extent is unknown. And in the case of the disease severity, the B117, there's some evidence that that variant is a little more lethal, but that's still the extent of it, the percentage we don't know. And then that's not established yet for B1351 and P1, okay? So, right, okay, and what I should say is that there was a recent study in which that showed very good evidence that antibodies from people who received the Oxford Astrazeneca vaccine and also those who received the mRNA vaccines, they are well able to neutralize P1, for example. As I said, not a problem with B117. So we're still in very good shape, okay? It's still worth getting vaccinated even when these things are present. So just to end, is there a way to prevent these mutations and to prevent new variants from arising? Well, the answer is yes. Since mutations only occur when viruses replicate within cells of an infected individual, the way to prevent these mutations and to prevent things is the same way we prevent SARS-CoV-2 infection. We basically prevent infections. So for COVID-19, that can be done by wearing your mask, social distancing, avoiding mass gatherings, staying at home when you're ill. We also have border restrictions in place, quarantine, all of these things prevent transmission, prevent replication, prevent mutations. As I said, the longer the pandemic goes on, the more chances of SARS-CoV-2 to mutate. So we have to double down on all of these things and stop this virus in its track. And the vaccines are a major part of that, especially since we're getting more and more evidence to show that they do reduce person-to-person transmission as well as protecting people against disease. And I'll end there. I hope I did not take too long. I'll stop my sharing now. Oops.
Dr. Carrington, Professor Carrington, rather, thank you so much. And I hope that our students had been listening, all of us, and have been writing down your questions. And I hope Dr. Carrington can stay with us.
Sure, of course.
Until our other presenter, Miss Dawn Marie Richards. She's still here with us? Yes, I think. Not seeing her. I'm not seeing her, Miss Walker. Ms. Richards?
Okay, perhaps you can let the students go ahead, or if anyone has questions, until we can get through to Ms. Richards, so that we're efficient with the time.
Fine, right. So while we wait, students, you can start sending your questions, because Professor Carrington gave an overview of what the viruses are like. She talked about transmission and the importance of wearing your mask and keeping social distance. And those are the key behaviors that we should have right now. Talk about the immune response. And many of you who did your microbiology and part of it, your epidemiology, you will remember this. We talk about the attackers, the B and T cells and the memory cells, right? Then she talked about the development of the different types of vaccines. One important takeaway from this is regardless of the efficacy, which they develop through the phase 3 trial of the vaccines, but regardless of the efficacy, all the vaccines so far are 100% protective against death, right? And it ranges, most of them are 100% protective against critical illnesses as well. So that's one takeaway that we should have. Then she talked about the different mutations and that the vaccines also are protective against those. So students, do you have any questions? Let us hear them. All right, so I have in the chat 2 questions, Dr. Carrington. One student asked, what causes mutation? And the other question is, do we currently have trials taking place in our population? So those are the 2 questions so far.
Okay, so the first one is what causes mutations, and the other one is trials. Yeah. Okay, so I'll answer the first one thing, and the answer is no. And I'm talking about the Caribbean now. We don't have any trials taking place in the Caribbean. As I mentioned, if you want to do a trial for a vaccine, you really want to do it in a place where you have lots of virus around, because you're looking to see if this virus, if the vaccine protects against infection. So doing it in, I mean, I know you'll have the situation in Jamaica is bad, but you need like 40,000 people to take, you know, in your trial, 40,000, 50,000 people in your trial. And at the time when they were doing these vaccines, our burden of disease in the Caribbean was extremely low. We would sit around, you know, for a very long time. We would not have a vaccine now if they did the trials in the Caribbean. They really needed to do them in places where they were expecting to see high transmission. So no. What causes mutations? So when a virus infects a cell, okay, one of the things we saw, the structure, it has this, it's sort of like a coat, and inside of it is this genetic material. That genetic material is delivered into the cell, and our cells have enzymes, and the virus sometimes they can make enzymes that replicate that DNA. So they copy that DNA or copy the RNA, make new copies of it. So it's an enzyme that does it, but the enzyme is not perfect, and the enzyme makes errors. All right, so sometimes it's copying ATCG, 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 and instead of putting a G, it puts a C. All right, so you get ATCG, ATCG, ATCC, and that's a mutation. Now, often when mutations occur, they're not compatible. The virus is no longer, it's sort of detrimental to the virus. I like to think of it as if you have a perfectly functional car, let's say the virus is a car, and you open the engine of that car and take out a part and replace it with a different part, all right? Do you expect that car to work better, to work just as well, or worse than before? I think most people would think that this car is not going to work. And that's what happens. You make these changes, these mutations, the vast majority of the time, you end up with a virus which is not able to replicate, it can't function. So it's detrimental to the virus. Sometimes you have changes that don't make a difference, and occasionally you have changes that will change the performance of the car, of the virus. Okay, so it's really an error that an enzyme makes. Any other questions?
All right, thank you. So far, I'm not seeing any other question. Do we have any questions from the group here among our own staff?
I always say if I don't get questions, either because I did a brilliant job or I did a terrible job.
I think you did.
One of the other. Brilliant or terrible. Brilliant or terrible. One of them.
All right, I have another one. What can you say about the side effects of these vaccines, such as blood clots?
All right, okay. So the Astrazeneca vaccine and the Johnson & Johnson vaccine have been, there's evidence to suggest that they may be associated with blood clots. Now, these are the adenoviral vector vaccines, and there's other ones as well. Now, I think what is the first thing is that the rate at which these blood clots occur, it's extremely rare. It is so rare that they're not even sure. I mean, it looks like it's related to the vaccine, but you can't be 100% sure. To tell you how rare it is with the Oxford Astrazeneca, it's 4 in a million, right? So for every million people, you might get 4 that get blood clots. Now, for every 100 people who get COVID-19, 16 will get blood clots. All right? So you are thousands and thousands of times more likely to get a blood clot from COVID-19 than from the vaccine. If it was me, I will take the vaccine. All right? So it's very rare. It looks like it does occur, but it is, I think the chance, it is about equivalent to the chance of you being, at least in the UK, there's a statistic I saw from the UK, the chance of being murdered in the UK, the chance in the Caribbean is probably much higher. So that's how common it is. And we still go out, people still go to the UK and don't worry about being murdered. It's also about equivalent to the chance of dying in a car accident in the UK. Probably more chance in the Caribbean of that, as I said, but so it's very low risk. The mRNA vaccines, I don't think they've seen much evidence of it there. And you have to remember as well, every medication, everything we do has risks. And when you think about vaccines, what you're thinking of is what is risk and benefits. The benefits of the vaccine far outweigh the risks, okay? Not taking the vaccine, think of that number. 16 out of every 100 people who gets COVID gets blood clots. 4 in a million people who get that vaccine get blood clots. So, you know, and the birth control pill, it's something like, oh gosh, I can't remember that number. Like 1 in 1,000. 1 in 1,000 women who take the birth control pill get blood clots. So it's vanishingly small. The risk is vanishing. It's there, but it's vanishingly small.
All right, I have 2 more questions, or 3 rather. Let me give them to you. What pre-existing conditions would be contraindications of the vaccines? That's one. And do you think we will need increased doses of the vaccine similar to the influenza vaccine?
All right.
And then one person wanted to find out, how does the vaccine in Cuba, how does it fit in?
Okay.
And will it be available and so on?
All right. So contraindications. So I always give out my disclaimer. I am not a medical doctor. So I am not somebody to come for medical advice. Okay. So, but as for the AstraZeneca vaccine, they say that people who have had not AstraZeneca, sorry. Well, any vaccine, I think all of them. Anybody who has allergy to one of the components of the vaccine should not take the vaccine. There is, in some countries, and to be honest, I don't know if it is the same for our region. In some countries, they're recommending that people who have a history of blood clotting disease might want to take a different vaccine. But to be honest, I don't know the recommendation in our part of the world. So I'm not a medical doctor. I leave that to your nursing people and the doctors and all that.
Okay.
Increased doses. I think that so as this virus evolves, you'll get you get slight different variants, and the variants are going to the vaccines are going to be the extent to which the vaccines protect against any disease is going to change over time. And so I do believe that going forward, and also we know from natural infections with coronaviruses that the immunity against them wanes over time. So you lose the immunity over time. So I think that we probably are going to need repeat vaccines. I don't know if it's going to be every year or every 3 years or what. But we've only lived with this virus for a year. We've lived with the vaccine now for a few months. So we don't know how long the immunity is going to last. We don't know, how fast, what's going to happen with the virus in terms of mutations going forward and stuff. But some of the vaccine companies are making variants of the vaccines to deal with the variants of the virus to make, to protect against all disease. So yes, and then the last one about the vaccine in Cuba. To be honest, I don't know very much about the vaccine in Cuba, so I will reserve comment on that. But I would be very happy if Cuba makes a vaccine and shares it with us because they have. So I don't know much about it.
Okay. Thank you so much, Prof. I see that Dawn is on again. Dawn, can you open your mic and talk to us a little before I introduce you to see how it is going with you?
Good afternoon, everyone. How are you all? Happy to be
fine.
here.
Dawn, have you changed your position? Do you think you can be on for about 15 minutes without shipping out?
I am hoping so. I have not reached home as yet. So
okay.
I am on the road. So I had actually pulled over, but I was doing fine, and then I don't know what happened.
All right. So Dawn Marie Richards is our public health nurse. She's a registered midwife, administrator, and she's a nurse advocate. She's an active member of the Kiwanis Club of St. George's, NAJ, and then she's a member of our Omega Kappa chapter of Sigma. She represents or has representation in the board as a board member of the Caribbean Nurses Organization, represents NAJ at local and regional levels, represents the Kiwanis at district and international levels, and she has, her saying is, do everything to the best of your ability. Mrs. Richards, we welcome you to present to us this evening on the COVID-19 vaccination scheduling and process in Jamaica and issues that are related to that. Thank you.
All right. Thank you for your introduction, Mrs. Anderson Johnson. Don't mind me if I say miss, because I'm used to that term.
Okay.
It's good to be in this forum at this time. I want to thank Professor Carrington for all that information that she just gave on COVID vaccines, the development, and just about everything, because people really ask a lot of questions, and sometimes we are not, we choose what we want to run with. So some persons are not really listening to the facts, or they hear the facts and they pick out the parts that they want to pick out. So we thank Prof for the clarification. A little on the vaccine process as we have it now. So in Jamaica, we would have started immunization using COVID vaccines as of the 10th of March this year. Our vaccine of choice would have been the Astrazeneca nCoV-19 coronavirus vaccine recombinant. Jesus have mercy, my battery dead. No, sir.
Ms. Richards, we are still seeing you online. Are you still there? All right, while we wait a little, Mrs. Chisholm Ford, our own resident public health nurse and student advocate. I wonder if I could call on you. Are you there?
Mrs. Anderson, Yes, I
I'm
did.
here, Mr. Johnson. Yes, I am here.
All right.
I did
Mrs.
ask her to be on standby.
Oh, you did.
Yes, I
Okay.
did.
So Miss Richards is gone off again. So Mrs. Chisholm Ford, we're going to invite you then to fill in for your colleague. Yeah, she said her battery died. Okay. To fill in for your colleague and to present to us, you know, the Ministry of Health, their process of rolling out this COVID-19 vaccine to our population and some of the issues that were related to that. And you maybe look at comorbidities and so on as well. Thank you.
Thank you, Mrs. Anderson Johnson. First of all, let me say I was not prepared to be on standby, but as a nurse, you have to respond to calls when you are called upon. So I will do what I can to the best of my ability. Thank you again. And as a member of Omega Kappa, that we are okay. I cannot put my camera on because I was not prepared. So the vaccine scheduling for the COVID-19 vaccine was done in 3 phases. It's for 3 phases. We were supposed to be doing the healthcare workers and all our first responders first, members of our military or police force, and following that we would have the elderly over 65 and persons with comorbidities, and then the third phase would be for the general population. However, due to many requests, overwhelming requests from the general public and several groups that were concerned, the Ministry of Health and Wellness relaxed the policy, and they included some members of the 65 and older with comorbidities in the first phase. So it was done, however, by scheduling appointments on the Ministry of Health's website, and that was a little challenging for most due to a lot of software issues as it relates to getting times, and some of the sites that were listed as vaccine sites were not yet ready. So persons who scheduled appointment for those sites turned up to the sites and were not able to be accommodated because the staff were not aware that they were supposed to be administering the vaccines. And as we all know, we have had several shipments of vaccines. Our last shipment came to us just about 10 days before they were expired. So the onus was on the Ministry of Health to get rid of the 75,000 doses within the time specified. And due to the efforts of all the public health personnel, including retired persons, we had midwives working, we had doctors giving vaccines. The 75,000 doses were utilized before they were expired. Our next batch is scheduled to be expired in May, and we should have been having a blitz that would have accommodated the general population starting this weekend, but that has been shelved due to unavailability of the vaccine, because as you heard about the COVAX facility, we are not getting our supplies as quickly as we would want to, because our more affluent partners in the north are, for want of a better word, hoarding. So we just have to take what we get. However, what we have, we are trying to reserve for the second doses, because the vaccine efficacy is not as vaccine is not as effective if you do not get the second dose. And the second dose should be given within 8 to 12 weeks after the first. So we are trying to ensure that as much as possible, we have vaccines available for the second doses. However, we are expecting to get another supply that should be able to cover the general population, those who are interested. I'm not remembering what else Mrs. Anderson Johnson had asked me to cover. I think I heard something about comorbidities. Is that right, Mrs. Anderson Johnson?
Yes.
What was it about comorbidities again?
Are there any comorbidities associated with, or what's the association of the vaccines with comorbidities? Yes.
Okay.
Mrs. Richards is back, so if you can fill in, Mrs.
She can
Richards,
take over, and we will partner.
Yeah, you can partner. You've heard some of the information that she's given already. Yes, you can fill in some of the things now.
Thank you, Sandra. Internet giving me a five.
That's how it is.
Ms. Richards, maybe if you turn your camera off, it's your, data may last a little longer. Yes.
All right. So I don't know how much Chisholmford has said, but
she went through the whole process of from the implementation of vaccination up to the last set of the blitz that went on.
Last blitz that we had.
Right. All right. So I heard you talking about comorbidities and so on. So we don't have any contraindications to the vaccine that we have, which is the AstraZeneca brand. But there are, as professionals, there are precautions that we would have taken. So like persons who are pregnant, you wouldn't give it to them, especially in the first trimester. There are some things that we look at. If the person is having a fever, we wouldn't give it to them. So there are some little things like that even though the research has not shown anything adverse in those persons, there are things that we still look at. It is not known to affect fertility. And as I said, the pregnancy risk has not been established, but there are precautions that we would take. As Chisholmford has said, we give it 8 to 12 weeks. Some places have been doing 8 weeks, some have been doing 10, but it can be given as early as 4 weeks. From as early as 4 weeks up to 12 weeks. The 75,000 doses to date we would have given us. Hello?
We're hearing you.
Okay. So to date we would have given over 80,000 doses across the island. I heard Chisholmford saying something about second dose, but I don't know what they're really asking us to do now is to give more persons a first dose. So it is us who are asking them to let us hold back and wait until that other set comes, because we don't know what is going to happen. We don't know how soon they'll be getting the new set. We have been told within the next 10 days, but we are not sure what is happening. For now, the schedule is, as of yesterday, up to the 30th of April, we should have resumed immunizing persons. But persons, unlike the Blitz Association, where persons could just walk in, they will not have that opportunity now. They must register on the government website. So the different areas have been asked to submit schedules to the ministry so that they can open the portal to get appointments. So persons will be waiting on that. Persons have started, some facilities have started immunization already, but we have from yesterday until the 30th to complete the quota that we have. The vaccines that some persons have, I know like for Northeast, the vaccines that we have won't expire until June. The remaining vaccines that we have won't expire until June, because that was the set that we got. And when the 75,000 doses came, they told us to hold back with those and issue the ones that would have expired faster. So all across the country, there are some persons have vaccines that have a little longer shelf life. We don't know what will happen with the next set that will come, but we work and we wait on the ministry and their directives. Just to say, as has been said already though, we have to have 2 doses for this particular type. It is not recommended that you switch types. That is not the recommendation. So a lot of us are hoping that the next set coming will still be AstraZeneca. There is a conversation about Moderna, some Moderna coming. But I guess some discussion will take place, but I know that some Moderna should be coming as well, sometime soon. I don't know if there are any questions in terms of side effects. When you know the regular side effects, that persons would have had: lymphadenopathy, decreased appetite, headache, dizziness. Some persons have flu-like symptoms, redness at the site, injection site. Some persons have nausea, vomiting, abdominal pain. Some persons have, I didn't see it in the literature, but persons complain of loss of appetite, and some persons complain of tiredness. So those are some things that I did not see in the literature for this particular vaccine, but those are the complaints that persons would have said to us, along with the other regular adverse side effects. I don't know if I have covered what you would want to hear, and if there's any other questions that you would want to ask.
Hey, thank you so much, Mrs. Richards. All right, so if they're the students, I'm not getting any more questions, but let us wait a little to see if they have any questions for you. Just to add that there are 2 persons who I know of who slept for like 2 days straight.
Yes, I know persons who, somebody told me that they had to go home from work. It's like it just came on them and they had to go home and go to bed, and they slept unusually long for the evening into the night. So yes, I know that is one of the remarks that persons have told us.
Okay, yes. All right, any other questions from this group? I don't see any coming from the students group. Any questions here? Okay, I have 2 hands up. Dr. Monroe.
Okay, thank you. Thank you so much, Prof. Carrington and Mrs. Richards, for your very informative presentation, and Mrs. Chisholm Ford for your emergency intervention that kept us alive. Thank you so much. So the question I have, Mrs. Richards started talking about it because she mentioned that with the second dose, they're trying not to mix types. And I was wondering, because that was a question that students had asked when we had a meeting with them prior to this. You know, if I got AstraZeneca now, and when my second dose schedule comes around, if AstraZeneca is not available, is that a problem? Prof. Carrington, is that problematic?
Well, I really don't have the information to tell you about, which ones can be mixed and matched. I know that in some countries they are allowing it with some combinations, and I know that there's studies going on to see how well they work. But I would not venture into there to tell you what works and doesn't. And I don't know the recommendations. I'm sorry.
Okay. So students, that is something that we would have to wait on. I know that when we asked the chief nurse that question, she said that she shared the same sentiments as Mrs. Richards, that she said, the aim is not to mix types at all, because that's out of an abundance of caution until we learn more about vaccination.
Yeah, I was just going to say that I think it's more to do with the mixing is more to do with not knowing the sort of efficacy at the end. These, they went to great lengths to do clinical trials to check how well these things work with, certain protocols and stuff. So you don't want to change the protocols. I don't think that, I don't think there's any sort of great risk if somebody gets the wrong second dose. But what we don't know is, how well protected will they be, So I think it's more about that than anything else.
Yeah, that makes sense.
Right. Thank you, Dr. Carrington. That's it, Dr. Monroe?
Yes, thank you.
Okay, great. So Ms. Brown.
Yes, thank you so much for your presentations. My question also relates to mixing, but in a different sense. Perhaps Mrs. Richards would be in a position to answer this one. I can remember very clearly the minister and perhaps the CMO saying at the beginning that their intention is not even to mix batches. So for example, if you got your AstraZeneca from India, for your second dose, they want you to get AstraZeneca from India and not AstraZeneca from Africa. Are we still maintaining that approach, or are we now just saying if you got AstraZeneca to begin with, it's AstraZeneca at the second dose wherever it came from?
All right, thanks for the question. The discussion continues, but I know that the literature for the AstraZeneca from India has asked that you get the same one. That's what the literature for the AstraZeneca from India. That one, the leaflet that comes in the vaccine box, the box, speaks to using the same one for the second dose. But as I said, yes.
I'm sorry, go ahead, and I will add something when we're through. I'm sorry.
Yeah, the discussion is ongoing, and the thought process is to stay with the same vaccine. That is a thought process. But the discussions continue.
All right, thank you. Mrs. Chisholm-Ford, you want to add to that?
Yes, thank you. I'm just adding that I'm agreeing with my colleague that we are staying with the same manufacturer, so that we are sure of what the reactions are. And just to add, when we are administering the vaccines, when you pull up from a vial, there are some leftovers in a vial, and we are told, we're also instructed not to mix from different vials. So whatever is left from a vial that does not make the 0.5 mils is discarded. So that is how cautious we are to ensure that you are not getting any mixture of the vaccines.
Okay, thank you, Mrs. Chisholm Ford.
Can I comment, Mrs. Anderson Johnson?
Yes, go
Yeah,
ahead.
I think what Mrs. Chisholm Ford just said and Mrs. Richards before her is really reassuring, because that's a concern that the students had, that we have the vaccines now. We don't know, some people have not been given a second date as yet, and you know how confident are we that we're going to be getting the same thing the second time around? And if we don't have that confidence, does it make sense even getting the first dose? And so this, what we're hearing from our public health nurses, is very very reassuring, I think, and the students should be reassured by the responses. Just wanted to make that comment. Thank you.
Yes. Oops. Thank you so much, Ms. Dr. Monroe. All right, I don't see any more hands. There's no more questions in the chat. And it is now 7:20, nearly 2 hours since we started. Dr. Carrington, I am so sorry, but when you're in Jamaica again, hopefully COVID will be over and we can have you out for tea.
Thank you. I'm half Jamaican, my mother is Jamaican, so half my family is there. Yes.
All right, okay, so next time when you're here.
Yes,
thank you
definitely.
so much.
Thank you.
Right, I want to thank Mrs. Garrix-Lloyd, and it was said already, but thank you, Mrs. Garrix-Lloyd. You were the one who, searched and found these wonderful presenters for us for this occasion. And thank you. I must say, the presenters, that you are very open, very knowledgeable. The presentations were done with clarity. And Dr. Carrington, I sat and I learned like a student. I mean, you went from the virus itself and you went through the whole process right to vaccinations. And I think I could understand very well. I made notes too. I can refer to them again. Mrs. Chisholm Ford, I want to thank you so much because, it is said that we should lay hands suddenly on no man, but here you were, ready because you're there in the field. And so you know exactly what was happening. So you were able to do that presentation and to help your colleague who was having difficulty. Mrs. Richards, thank you so much. Please make sure that you are very near to home now, curfew time, because you said, we saw that you were driving, you were in your vehicle. I mean, the kind of commitment that you showed this evening in just coming to us to make this presentation. Thank you so much. We have learned in this group, and I am sure that our students have learned much and have benefited. Our two objectives, I am sure, were met. Increased knowledge of COVID-19 vaccines and understanding and appreciating the process of the rolling out of a new vaccine in Jamaica. Thank you all so much. And we hope we have another session like this again. Dr. Monroe, yes, thank you. Yes, thank you, Mrs. Ashman, for being there, doing overtime for us in this session. Thank you. All right.
Thank you very much.
Yes, thank you. So we'll call on you again.
Thank you for having me. And I will make myself available as much as I can.
Wonderful, wonderful. That's the heart of an Omega Kappa member, yes.
Yes.
Yes. Thank you all. Bye-bye.
Bye.
Bye, everyone. Bye.
Thanks, everybody. Thanks, Purple Heart. Thanks, Prof. Dawn Marie, good to see you.
Good to see you too, Dr. Monroe.
Yes. Thank you, Sandy. Take good care of yourselves. Shay, please get home safely.
What do you mean get home? Shay is still at the office?
Yes, she is.
I
because
am
she wants
thinking that she's doing this from home.
to know she stayed.
No, she's not. She stayed.